Hello, everyone, and welcome to Beyond Modulators, Genetic Therapies on the Horizon at ResearchCon. My name is Julie Desch, and I'm a 59-year-old person with cystic fibrosis, and I live in the San Francisco Bay Area. I'm moderating our session this evening, and I'll be introducing our other speakers in just a minute. But before we get started with this session, I want to go over a few items related to the technology and how we can all interact. We'll be taking audience questions during this session. So if you have a question, you can submit it at any time by clicking the icon on the right side of your screen labeled Q&A. You'll also see other questions there, and if you give a thumbs up to the questions you like, they'll be upvoted to the top. When thinking about your question, please formulate it in a way that will be helpful to everyone here. So we won't be able to answer questions about individual diagnoses or treatment plans. We encourage you to use the information you learned today to have conversations with your care team. Otherwise, we'll do our best to answer as many questions as possible. You'll also notice there's a chat bubble on the right side of your screen. This is the group chat for everyone that's in this session. You can open it or hide it by clicking on the icon. If you have a question for the speakers, make sure you use the Q&A icon to submit it, not the group chat. And if you have any technology problems during the panel, please go to the Tech and Support room on the home screen of the event, which looks like a lab, and you can get help there. Now I'll introduce the rest of the panel and let each person introduce themselves. So first to Sharon Cray.
Hi, everybody. My name's Sharon Cray. I am from Mount Laurel, New Jersey, which is just outside of Philadelphia. I am a CF mom. I have two children. My oldest is a healthy son, and my youngest is a 23-year-old CF young lady who is working on a graduate program right now. We're really proud of her. She started Trikafta in November and is doing great. We told her she could get that last semester of her graduate program done, healthy as anything, and then COVID hit, so that hasn't worked out that well, but she is doing really well. Our speaker tonight is John Sheridan, who you've probably met along the way tonight. He's one of our co-chairs. I will turn it over to John.
Great, thanks Sharon and Julie. Yes, you've probably seen me already as one of the ResearchCon co-chairs. So just a little bit of background about myself. I am the Director of Research here at the CF Foundation. In that role, I primarily manage the academic research programs that are supported through our Path to a Cure initiative. So these are programs that are focused on gene therapy, gene editing, stem cells, nonsense therapies, a variety of different approaches. So I'm really excited to be here tonight to really give you a bit of a more in-depth discussion about some of the genetic-based therapies that Bill raised during the keynote session, as well as cover a couple others that maybe weren't brought up in that keynote. So let me go ahead and share my screen. Just 1 minute. Okay. There we go. Great. So you've already seen a version of the slide in Bill's presentation, but really I want to start out with the goal of the CF Foundation is really to have a cure for all CF patients. Now, as Bill mentioned, the past 50 years have really been focused on treating the complications of CF, the lung, the livers, the pancreas, the gut, a variety of different issues, and these are given continuously throughout life. It wasn't until recently where we actually now have therapies that can specifically target the protein, as in the CFTR modulator drugs, and these are given daily. But as we're thinking about how we can actually get on that path to ultimately cure CF, we need to be thinking about what are the genetic-based approaches where we can correct and cure CF. So the first approach that we'll be talking about is really around the RNA approach. And this a therapeutic approach wouldn't represent a permanent fix, but it would potentially represent a periodic therapy where you may only need to receive it once a week, once every 2 weeks, a variety of different approaches. And then really to get to a permanent fix, a permanent repair, is really when we're looking at the gene level, looking at the specific CFTR mutations and potentially correcting that mutation. So I think it's important to really get everyone on the same page, as some of the topics we're discussing can be a little complex. So we have in biology what's called the central dogma. And this is essentially that DNA leads to RNA, which leads to protein. So you can kind of think of this using an analogy that DNA is the blueprint. It's the instructions. It contains all of the information you need to build that protein, the final product. In this case, it's a house. CFTR is represented by a house. So the DNA, the blueprint, you make a copy of it. And that's what the mRNA is. It's the instructions that then those workers can take to the building site to build the protein, in this case, CFTR. So it's important that we can target the CFTR gene, mRNA, and protein, along this way at various different steps. And while a lot of work have focused on the CFTR protein itself, we're wanting to start thinking about how we can focus on the DNA and RNA level. So before I go into the rest of the slides and more of the information, we have a brief video. It's about 2 minutes that will go over a couple of these in a little more detail. So I will stop sharing and we will play the video.
Gene editing would repair CFTR mutations in the person's own DNA. There are multiple gene editing techniques, including CRISPR-Cas9, zinc-fingered nucleases, as well as others. Each of these techniques remove the mutations from the gene. To fix this damage, the cell puts in the correct DNA sequence for a functional CFTR gene. Gene editing can be done with cells inside the body, or in cells that have been taken out, corrected in the lab, and put back into the body. Gene editing can either be tailored for each specific CF mutation or designed to correct many or all mutations. Gene editing changes the DNA permanently. If CFTR mutations in stem cells can be edited, all future cells derived from these stem cells will make correct CFTR protein, making repeated treatments unnecessary. One possible way to restore CFTR protein is gene transfer. Gene transfer would work by inserting a normal CFTR DNA sequence into the cell so that it can make functional CFTR protein. Gene therapy could be inhaled or given through an IV. However, getting DNA into lungs is challenging. Cells are surrounded by protective membranes that are designed to keep out foreign objects. And in the lungs of people with CF, cells are also covered with a thick layer of mucus. Specialized delivery mechanisms called vectors are being developed which would take the new correct CFTR gene into the cells. There are multiple types of vectors derived from viruses or synthetic molecules. Gene transfer could potentially work on anyone with CF, no matter what kind of mutation they have.
Okay, so what that video showed us was a really a brief overview of kind of what gene editing is and what gene transfer is. It didn't go into mRNA therapies, which is what I'll be going into next. So if we move forward, sorry, 1 second. Having a bit of there we go. So if we think about what's in our therapeutic toolbox, we'll be first looking at kind of the RNA therapy approaches. Now, there are several different approaches, and Bill mentioned mRNA as well as antisense oligonucleotide therapies in his keynote address, but he didn't talk about tRNA, which I'll be going into a little more detail in this talk. So those are some of our options of what the RNA-based therapies are. Now, who actually will benefit from these therapies? Really, when we're looking at mRNA therapy, we think of that as really a mutation agnostic approach where we're just providing the full sequence of CFTR as mRNA, and your natural enzymes and processing will translate that mRNA into protein, and it doesn't matter what mutation you have. Antisense oligonucleotides really are probably going to be more applicable to splicing mutations, and I'll go into a little bit more detail on that, as well as then tRNA therapies are a unique RNA-based approach, and those would likely be more applicable to nonsense mutations or patients that have a premature termination codon who don't ultimately produce protein. So let's start with mRNA therapies. This is the one where we've actually have seen a clinical trial be completed with an mRNA therapy approach. So this was done by Translate Bio, and I want to start with the caveat that this was a very short trial. This is 8 days maximum with a small population of people. So really, these are very preliminary data that they've released last year. So what they did is they packaged mRNA into a lipid nanoparticle, which is composed of fats, and then delivered that via nebulization to patients with CF. And so they did it at different dosages and then looked at lung function at different days. And what they found is that when they, over time, they could see an improvement in lung function. So that's really interesting and exciting results. But as I said, it is preliminary, and there are other companies that are working in this space. Bill mentioned Arcturus is another one that is also developing mRNA therapy. But really, we think we're really excited about an mRNA therapy approach because it would allow us to get the quickest way possible to an actual therapy for all 100% of patients. So let's talk about tRNA therapies a little bit. So tRNAs are a unique type of RNA. So what they do is they are actually responsible for helping translate that mRNA sequence into protein. So you can kind of think of this as a tRNA is like a delivery person, and that person carries a package, and that package is the amino acid. And when those amino acids are put together in the chain in the very specific order based on that mRNA sequence, it then creates the protein of CFTR, which folds correctly. So in the case of someone with a nonsense mutation or who has a premature stop codon, a tRNA comes in, recognizes that stop codon, and doesn't transfer another amino acid to that growing chain, and therefore terminates translation of that mRNA. One potential option to treat this is to utilize an engineered tRNA. And this is a specific type of tRNA that could recognize that mutation of, that premature stop codon and then transfer an amino acid, allowing you to essentially bypass that amino acid and proceed translation down the mRNA therapy. So this is a very early stage work, but we are very excited about it. It's a unique way that might address some of the specific challenges of targeting nonsense mutations that aren't necessarily a gene editing approach. So moving on to the antisense RNA and oligonucleotide therapy, which is a mouthful. We usually just say ASO. And what these are is they would allow aberrant splicing of a mutated CFTR mRNA to splice normally. So when we have the mRNA sequence, there's actually sections of it that get cut out before it's actually translated into protein. This is called pre-mRNA, and we cut out, in this case, there's these little white sections that get cut out. What happens is if there's a mutation in one of these white sections, splicing doesn't occur normally and kind of could jumble up the entire mRNA sequence, which therefore leads to a non-functional CFTR protein. An ASO actually would be a small sequence that would specifically hone in on that mutation and effectively block that splicing event from occurring and allowing normal splicing to occur. So this would lead to normal mRNA and normal CFTR protein. So one common mutation that this may be applicable to is the 3849 plus 10kb mutation. And there are also some companies that are also already working in this space. So let's move on to some more genetic therapies. So we'll start with gene transfer, and then we'll talk a little bit more about gene editing. So gene transfer therapy is the process of adding a normal CFTR gene into the cells of a person with CF. So this doesn't actually replace a mutant CFTR gene. It's essentially just adding another copy. So if you think about it, you would really ultimately have 3 copies of the CFTR gene in any given cell. And that's fine because we know we just need one normal copy of CFTR to have normal function. So gene therapy really has advanced quite a bit since the 90s and early 2000s. I think when it really took off in the CF space. And obviously we didn't have much success there. But we now actually have a number of FDA-approved gene therapies that have come out, really focusing on oncology, beta thalassemia, which is a blood disease, retinal blindness diseases, as well as one called spinal muscular atrophy. And spinal muscular atrophy, or SMA, is a really devastating disease where most patients who are born with it don't survive many years at all. And so when a drug was actually approved for this, a gene therapy, it was actually remarkable that something like this came on the market. And so what I want to do in the next slide is kind of compare how an SMA gene therapy works versus how a CFTR gene therapy would work. Because while they're similar, there are noticeable differences that may make CFTR more difficult than SMA. So if we first look at how we're actually going to package that gene to deliver it to the airways or the cells of interest. So the SMA gene itself is very small. So it can be packaged into the vector, which is typically a virus vector that's essentially just a shell of a virus that can be used to package material. It can fit in there. But the CFTR gene is very large. And so what happens is it exceeds that cargo capacity of that vector. So we have to think of different ways that we could package the DNA or the gene into the vector, develop new vectors, make some changes to the DNA in a way that we can package it appropriately. So that's one challenge. Another challenge is that delivering something, in this case, we'll just focus on the airways, can be very difficult. So targeting the airway epithelium through nebulization can be challenging. You have to navigate the mucus, immune cells, and get to the appropriate cells in the lung. So in SMA, it's different in the sense that this is an injection that's directly injected into the spinal column. And it hones in on neurons, which is where the defective protein would normally reside. And so there's an easy access point to those cells that need to be targeted. Another issue is that neurons are incredibly long-lived, so they don't really replicate. And so if delivering a gene therapy to that cells means you likely would never need to redose those patients again. In CF, we would likely have to redose patients with the gene therapy because it's usually not integrated into your genome. So it doesn't always replicate as cells divide. And we know that the airway epithelium is active and cells divide and repopulate the airway epithelium after damage. So that's something that we need to think about, is how do we actually redose as well? So let's now talk a little bit more about gene editing. So this is an approach where we actually could repair the defective gene in your own DNA. So this typically involves some sort of enzyme that's targeted to that gene. Now, you've probably heard about some of these different approaches. Really, CRISPR-Cas9 has really taken over. Everyone seems to know CRISPR-Cas9, and it's a really impressive technology. But there have been other ways that could be used to target and edit the gene, so such as zinc finger nucleases, which have been used in some clinical trials, ongoing clinical trials. And with that, we don't actually have any approved therapies using gene editing at this point. However, there are a number of clinical trials ongoing. So we definitely think we will see gene editing therapies become approved by the FDA. CF likely will not be the first one. Likely, I would think potentially a sickle cell or a blindness approach would be more of the likely an early gene editing therapy to hit the market. Now, I want to compare kind of the CF mutations to sickle cell. In this case, sickle cell is caused by one mutation, whereas in CF, there's 1,000+ mutations. Now, this creates a unique challenge because most gene editing approaches rely on a gene editor and a mutation-specific guide. So these are guides that hone the gene editing machinery to that specific mutation, which is really important because you don't want off-target editing. You want to edit the specific mutation. So what that means is, if we could develop a therapy that targets, say, the W1282X mutation, that won't necessarily work for the G542X mutation. So we may need a separate therapy to target that mutation. Now, there are a number of ways that individuals are looking into that would allow to correct more mutations, all mutations. And if anyone has questions, we can discuss that a little bit more in the Q&A. But there's a lot of ideas and concepts out there that might allow us to target more than one mutation at a time. But that is a real challenge that we are facing. The other issue that we need to think about for gene editing is, if we're going to have a permanent cure in the airways and really anywhere else in the body, is you need to target the progenitor cells. Now, in the airway epithelium, this is a set of cells which are called the basal cells, and they really reside in this protected domain in the airway epithelium. They're at the kind of the base, the bottom of the airway epithelium, and they're protected there because we don't want them to get damaged. If you get a viral infection and your airway epithelium is stripped off, you need those cells there to then repopulate the airway epithelium. But what that means is actually developing a vector that can specifically and efficiently target those cells can be a real challenge. So there's options. We could potentially come from the bottom, which would be more of an IV via the blood circulatory system to deliver to the cells, or create unique delivery vehicles that could penetrate through that airway epithelium and get to those cells in order to edit. And so that would represent a permanent one-time fix in the airway epithelium if we can do that. So there's a lot of work really going on in all of these spaces. We have numerous academic research programs that are focusing on a variety of different approaches, as well as several industry work ongoing in these different areas. So really, one of the big roles of the CF Foundation is to really build the science. So really, we're supporting a lot of different groups in this. We have an epithelial stem cell consortium. We funded several different groups on gene therapy and gene editing. We provide a lot of resources to the community and industry that will hopefully enable them to advance these therapeutic strategies. And really, ultimately, our goal is to get to 100% where everyone has a therapy that prevents disease. So with that, I will be happy to take questions. Let me just stop sharing.
Okay, thank you, John. That was excellent. Kind of the obvious first question is, what's the timeline for this? When can we expect to see these different genetic therapies in clinical trials, and then ultimately reaching the clinic?
Yeah, so Julie, this is definitely, I think, the top of the mind for a lot of people is when are we going to have these therapies? And the good news is at least the mRNA therapies have actually advanced to a clinical trial. While it's one company, that's really exciting news that we've actually seen that in a clinical trial. So I think likely mRNA will be the first approach we would see advancing to hopefully to patients, but I would put a gene transfer therapy as close behind that. I think gene editing is further away because there are a lot of unique challenges associated with it, but we really need to do a lot of the basic research now to enable that technology to become a reality.
Thanks, John. Another question we have for you is, when will these genetic therapies be able to be more systemic and not just focused on the lungs?
Yeah, so we've talked a lot about targeting and focusing on the lung. We recognize that CF is a whole body disease. It doesn't just affect the lung. But one of the challenges we have in developing these therapies that really target specific cells is it becomes a real challenge to use the same delivery vehicle to target multiple tissues. So, our goal ultimately is to target everything. But I think we're initially focusing so much on the lung because that's where most of the mortality and morbidity comes from, is the lung. So we certainly want to and will continue to fund research that looks beyond just the lung.
Excellent. Okay. What are some of the potential off-target effects that you were talking about? And what are the risks of those associated
Sure.
with genetic therapies?
So that's likely the off-target effects of gene editing, and that's a real concern. So we need to make sure that a technology is safe and isn't going to edit in an area it's not supposed to edit in. So the good thing is that CRISPR and some of these other approaches are highly specific. You get a lot of DNA damage and breaks from flying in an airplane, from solar radiation, and your body repairs that DNA damage you receive. So that's a normal process, a normal repair process that your body has. but we want to make sure, that we're not actually editing and changing. So there is a real concern that you could induce a off-target edit, but I think the chances of that are low, but certainly we need to explore that further. They're not low enough where we don't have to be concerned about it.
Thanks, John. I had a question about can patients stay on modulators when we go to clinical trials for these other types of therapies?
That's a very good question. I think it's going to depend on what the therapeutic approach is. So for an mRNA therapy, you likely, I believe I think the Translate Bio trial that they accepted patients who were on modulators. So I think that's not a factor for necessarily an mRNA approach. I think when we're talking about gene editing, it's likely going to be first targeting some of the patient population that doesn't have access to modulators. Really the patients on nonsense mutations or other rare mutations. Really, because I think early on, these are likely going to be very targeted therapies and not a broad therapy. So it may only be applicable to a subset of patients. So we think likely that will be the first patient population that would be approached for a gene editing therapy.
Great. And then I have an audience question here. How will genetic therapies work for rare frameshift mutations and missense mutations? And then there's another question sort of related to that. For nonsense mutations, we're working on read-through therapies. For splice mutations, we're working on RNA therapies. Does that make class 1 frameshift one of the most difficult to fix?
So that's a good question, and I think it's not necessarily that a frameshift mutation wouldn't be able to be corrected via gene editing. How usually gene editors work is they cut the DNA, and a portion of that DNA then gets kind of degraded away, and you provide a template that then your natural enzymes that copy DNA when your cells divide come in and copy that template and fill it in and correct that mutation. So you could correct that frame shift as well. So it's not that those would be necessarily harder to fix. Another approach that is being explored a lot at the very early academic research stage is what's called a super exon. And this is where we could potentially take a large section or the entire CFTR DNA sequence and using gene editing, put that in front of your current CFTR gene, and would still utilize the same mechanisms and machinery that normally runs that gene, but would essentially correct a majority or all CFTR mutations. Now it's a huge amount of DNA to insert, but it's something that we think is ultimately a strategy that will correct all or most mutations. It's a bit complicated, and I understand that, but it's a really interesting and intriguing approach.
There have been several questions on the topic of stem cell therapies for cystic fibrosis. Can you talk a little bit to that, John?
Sure, and I didn't really cover stem cell therapy in the presentation, but this is an area that we are very much focused on. We are supporting a epithelial stem cell consortium, which is composed of roughly 10 leading experts in the stem cell field. And so this group, part of their job is to, one, identify what the appropriate stem cells are in the lung initially, and how we could target those cells. What are the markers that would allow us to effectively deliver a gene therapy or a gene editing vector to those cells? Another aspect is that they are trying to devise ways to create induced pluripotent stem cells. So these are cells that they can take from skin and reprogram them to turn into airway epithelium. And the other aspect is engraftment. So a stem cell-based therapy is very different than some of these other therapies we're talking about. There are similarities, but there are differences. So, for example, if we take cells from a patient, we create pluripotent stem cells from those cells. We could correct the mutation using gene editing in that cell population, and then re-deliver those cells to the airway epithelium. But engraftment of those cells is probably the biggest challenge there, because your airway epithelium is really a tight, closed, protected area, and it's not easy just to throw in cells and expect them to stick and engraft into that airway epithelium. So there's going to have to be a lot of work in how do we open up the airway epithelium in a way that could receive those cells? Could we implant something that on a scaffold that would then enable them to stay and repopulate the airway epithelium? So there are a lot of additional alternative challenges associated with a stem cell-based approach. But, it's an area that we are really actively pursuing.
Another audience question. What clinical trials, I know you mentioned that one small trial with mRNA that was inhaled, but have there been any others with gene therapies?
I mean, there were numerous gene therapy trials back in the day, in the 90s to early 2000s, a variety of different phases that all essentially failed. And from that point on, there really haven't been any gene therapy, gene editing, trials. So really, we're essentially re-emerging with the gene therapy, and gene editing is so new that there just really haven't been any trials. We haven't developed the technology to a point where we can implement it in a therapeutic fashion. So really, mRNA right now is the first one we've seen in a while, but I anticipate there'll be more.
A member is asking about the difference between the three companies, Translate Bio, Arcturus, and I'm probably murdering those names. What is the difference between the three companies that are in that space and the research that they're doing?
So, I don't know the details of the companies, exactly what their proprietary information is, but I do know that Arcturus and Translate Bio are using similar approaches where they're using a lipid nanoparticle and delivering mRNA. How they do that may be slightly different. Some of the composition of how they design that vector, that lipid nanoparticle, may be different. And the third company is Acuitas, and that's a company that we really have just very early on discussions with them to do some initial screening. They have a library of a variety of different delivery vehicles that we can test, but that's very early stages with that company.
Okay, we have another audience question. Is there conversation happening about access to these clinical trials? Thinking about those with rare and nonsense mutations being a small percentage of an already small population of people with CF.
Yeah, that's definitely a real concern and a real challenge. We think when it comes to clinical trial design that we're going to really have to rethink how we conduct a clinical trial. We're not going to be able to have a large patient population enrolled in one of these trials. So how do we accurately measure a positive effect that's statistically robust enough that we can rely on and convince the FDA that it's an effective and safe therapy. So that's something that we have been discussing quite a bit within the foundation between our research affairs team, which I work with, and kind of our therapeutics development team, which is led by JP Clancy, to think about how do we develop in the preclinical space appropriate tests and endpoints to then decide to move something to a clinical trial. And then what are the appropriate endpoints in that clinical trial that would convince us that we're seeing a real response? So it's something that is ongoing, and it is a challenge, but I think it's something we're really focusing on right now, making sure we have that in place when the time comes.
Another audience question: how do the read past the stop codon tools not screw up all the other proper stop codons on all the other proteins in the body?
So I can't say for certain I know every detail about how that would work. I don't know if it really truly is known at this point. Now, that isn't to say that there are differences in a sequence with a premature stop codon, the environment it's in, compared to a normal stop codon. There are differences with the mRNA that the machinery that's translating the mRNA can recognize. We also have a process, everyone has a process called nonsense-mediated decay, or NMD. This is a process where your body, your cells, recognize that there is a premature stop codon in mRNA sequence. It recognizes that and degrades the mRNA because it knows it's defective. So that's a challenge is with a nonsense mutation, is the abundance of mRNA is lower as well. So we need to have a way to increase the mRNA abundance level and ensure read-through of the mRNA. So that is something that will certainly be a major safety evaluation and looking for toxicity, is that you're reading through normal stop codons, because obviously that would stop a therapy from being developed.
Yeah, that could be disastrous. Okay, another audience question. Are there any in utero stem cell therapies being looked at, or is that too far down the road?
I think something like that is very, very far down the road. Certainly with the stem cell approach is even further, I would say, down the road. Right now, we will likely have to be focusing in adult patients based on historical work with the FDA and how they approach these type of therapies. So that obviously creates a challenge, because adults with CF likely have more lung damage, more mucus obstruction than a child with CF. So we have to create an approach in therapies that can effectively treat an adult with CF before we can get to the child with CF. So, I mean, it'd be wonderful if we could correct it in children, but the way the FDA works, it's highly unlikely that we would be going that route.
Here's a question. Will there be a focus on the DELA508 mutation in gene editing because of the larger population, and it will reach the greater population it will reach, or is there an easier place to start? And I guess along with that, is there a difference between editing for one mutation other than another mutation? Is one more complicated than another?
Yeah, so one of the reasons we don't think we'll be focusing on the Delta F508 mutation initially is the fact that there are modulator therapies that are highly effective. So we don't think the risk of editing that mutation would outweigh the benefits that you currently have with the modulator therapies. So we think that our initial focus will be on those patients that don't have access to a therapy that directly modulates the CFTR protein. So I think a lot of companies that approach us, at least, they all come with Delta F508 initially, and they may focus on that. I mean, we work with them, but I think there's real concern that you would be going after a mutation where there is an effective therapy for. And I also wonder what the ability to recruit patients would be for that. Would a patient be willing to potentially change their genome when they have a current therapy? So it's a difficult area to think about, and it's a challenging area when it comes to figuring out what is the appropriate population to be focusing on. Good question.
Here's another audience question. I read a study some years ago that indicated communication of correcting genes in cells adjacent to targeted cells and indicated benefit communication on a subcellular basis. Has this been seen in gene therapy studies that you know of?
So I don't know if I understand the question entirely, but it kind of sounds like you're potentially talking about exosomes and cell-to-cell communication. Because one thing that we do know that epithelial cells, as well as a lot of other cells throughout your body, is they produce a lot of these they bud off exosomes, which are they're kind of like a lipid nanoparticle. They're just a little piece of membrane. And these can contain things like microRNAs and other information that can fuse with other cells. And this is a way that you can shuttle information between different cells. But I don't think it's ever been shown that you could transfer a full CFTR mRNA between cells. Usually these are very small pieces of mRNA or small proteins that may be able to help regulate some genes. So I hope I'm answering the question right. I'm hoping I understood that correctly, but I don't think we're seeing anything in that area right now that would fully apply here.
John, will transplant patients be able to take advantage of these different genetic therapies?
Yeah, I mean that's a really interesting question, because while we have been talking about lung focus, we do recognize that correcting the lungs does not represent a full cure. And someone that has undergone a transplant still has all the other issues. So we certainly would like to see, potentially an mRNA therapy or a gene transfer therapy that could be delivered systemically and target the appropriate organs and tissues. One of the concerns about doing a full systemic, delivery of an mRNA is that we would deliver it to tissues that don't normally express CFTR. So we don't know what the full risk of having CFTR expressed in a cell that normally doesn't express it. So that may be less of a concern with a gene therapy, because you would have the normal genetic environment that regulates expression of that gene, but an mRNA therapy wouldn't necessarily. So there wouldn't necessarily be the proper way to regulate expression of that mRNA or translation of that mRNA. So I still think, at least early on, we'll be targeting specific tissues, even beyond the lung.
Okay, here's a timely question. There is so much talk about viruses in the news these days. Is it safe to use a virus as a vector?
Yeah, so I think this is a common concern about viruses. And when we talk about gene therapies, we're always talking about viral vectors. So there's really a couple different ways you can deliver material or genetic material to cells. It's either a nanoparticle, frequently a lipid nanoparticle, or a viral vector. And so there's a lot of different types of viruses that are used. And essentially what these are they're just the husk of the virus. So there's no actual coding information for the virus to replicate. So one of the most common viruses is the AAV, or the adeno-associated virus, and these have actually been approved as therapies. So the therapy for spinal muscular atrophy is a viral vector delivery. So we know that these can be safe. There's been numerous studies with them, and that there really isn't a risk of much risk of there's always risk of an immune response to these, of course, but there's no risk of replicating a virus and having a viral infection spread throughout the body, because they simply don't have the appropriate DNA or information in the virus to do that. So they're really just a shell.
Here's an interesting one. Are these inhaled therapies likely to be dangerous to those who don't have CF that are in the room?
That's a very intriguing question. I think it depends on what approach it would be. I think an mRNA would have very low risk. I think actually inhaling off from someone nebulizing a therapy and getting it to go to the location in the lung would be very, very low risk. I think some of like a gene editing therapy, that hopefully would be a 1 or 2 time thing and would be corrected. So that probably wouldn't be an at-home therapy that you would really be receiving. It would probably be done in a clinic somewhere. But we're so far away from actually thinking about that unfortunately I don't have a solid answer for you.
Another audience question. Are there any animal models of CF disease with the Delta F508 mutation, therefore allowing initial studies to not depend on CF patients' willingness to enter the gene therapy, editing, or gene transfer research?
So there are numerous animal models. So CF is fortunate in the sense that we have so many animal models. So we have animal models that have specific mutations, animal models that have the human sequence with the specific mutation. There's certainly Delta F mutation animals. There's mice, there's rats, there's ferrets, there's pigs. There's even sheep and a variety of other animals that either have had CFTR knocked out or they have specific mutations. So any real therapeutic development program is going to require testing in an animal model before the FDA would allow it to be entered into a human clinical trial. So we certainly would want to utilize that, but we also have, what's great is that, we've set up a system where we can actually acquire explanted lung tissue. And so that is incredibly helpful to the community, because when we get explanted lung tissue that carries some of these rare mutations, that allows researchers to evaluate that in actual human samples and human tissue with all the conditions that a patient would have. So those are incredibly valuable, acquiring explanted lung tissue. So a variety of different approaches would be utilized.
A combination of a few questions I see. Why are other diseases like sickle cell and blindness, HIV, and a few others ahead of CF in effectively utilizing gene therapy? Gene editing.
Editing. So one of the reasons is simply access to the tissue. So for sickle cell is you can take bone marrow out of a patient, and those are the stem cells that create all your other blood cells. So you can take that bone marrow out of the patient, do your editing in a culture, in a petri dish, and then reintroduce that to the patient. And because it's bone marrow, they will home into the bone marrow, the same way you would do a bone marrow transplant, and repopulate that, and essentially then correct it. So they take the barrier of delivery is a much less of a barrier than it is for CF. Now for blindness, what's very good about using these for an eye disease is that the eye is generally considered immunoprivileged, meaning it's a self-contained system. And so there's not a lot of other outside influence that gets into it, and you can simply inject the therapy into the eye. And so it's delivered right to the location where it needs to be.
Ow.
I know, it's a little scary, but you don't actually have neurons to feel pain on your eye. So you don't want to actually really feel some pressure.
I'll have to take your word for that, John.
Yeah. Yeah. Okay, so we're down to the last question here, John. And this is a good one. What is the CF Foundation doing to support these genetic therapies?
So really, I think our whole research focus has shifted so much from focusing so much effort on developing modulator therapies to genetic-based approaches, because that's essentially what the Path to a Cure initiative is all about. So we're dedicating a lot of resources and energy to developing these therapies. So it is really our right at the top of the list of priorities is finding great researchers, scientists, companies that are developing these technologies and advancing them.
Excellent. Okay, so we're just about out of time, so I want to wrap up by thanking, first of all, John and all of our speakers for sharing your knowledge and insight about genetic therapies. And thanks to all of you for your attention and your questions. There are many wonderful resources related to all of the event sessions in the resource room, which you can access from the homepage of the event, which looks like a lab. For this session, you'll find the slides that we shared and other items the speaker thought you might find valuable. Please spend some time checking out the resources and download anything you want to keep. We're now going to have a 5-minute break, and then we'll be congregating in the keynote room for a short closing of the event. Take a moment to think about your favorite part of ResearchCon, because you may have the chance to come on screen and share it with everyone here. So now you can close this window and return to the ResearchCon environment. And thanks again for coming to learn about genetic therapies on the horizon.
Thanks, everybody.
Thank you, everyone.
Bye.